Now, this particular section, we are still back on the specific immunity, and we will now look at the cell-mediated response, which is a lot more complicated than the antibody-mediated response. Recall again, the antibody-mediated response primarily was played by the B cells. How does the cell-mediated immunity works? Now, cell-mediated immunity is mainly used for killing infected cells, in particular cancerous cells. So they're very unique in its antigen recognition sites. And there are many players involved in this particular process. There is the first one we want to look at is the helper T cell, also called as the CD4. They recognize antigen that has been processed and is fragmented on the surface of the antigen-presenting cell. So we will talk about the antigen-presenting cell in the next slide. But the CD4 or the T cells are the cells that will recognize the antigens. Now, once these cells recognize the antigens, they actually release certain signals, and these signals are then are looked at, or they're attracted by the cytotoxic or the CD8 cells that will come and kill that target cell. So having a helper T or CD4 attached to an antigen gives it a signal, so the cytotoxic or the CD8 cells will come in and it will either touch kill it, or it will release a certain chemical called as perforins, and the perforins will then destroy the cell. So CD8 or the cytotoxic cells are the ones that will destroy the target cell that are infected or cancerous. However, during the formation of both CD4 and CD8 cells, there are some cells also called as the memory cells formed, and these memory cells will facilitate the contact for further use. So, for example, if the same antigen was exposed later, the M cells will facilitate the recognition of that antigen. There are also cells called as the suppressor T cell or the regulatory cells, and they regulate immune response and maintain its balance. In other words, if accidentally one of our own good cells have that antigen, then the antibody should not destroy that. So that will actually suppress autoimmunity if the T cells were not there. So they combat. So our own good cells are not killed. Antigen-presenting cell, I'll show it to you in the next slide. And the natural killer cells, we'll also talk about that in the coming up cell-mediated toxicity slide. So again, this particular slide gives you all the key players that are present in this process. What are antigen-presenting cells? Now, antigen-presenting cells can be either dendritic cell, a B cell, or an activated macrophage. What an antigen-presenting cell does to this T cell is it recognizes the infected cell and attaches to it and releases the MHC2. Remember, I said earlier before we did that in the antibody-mediated that the B cells were attaching to an antigen and releasing a signal, which is the MHC2 protein. Now, the MHC2 protein will then help the T cell to recognize that infected cell. So it's actually a self-check mechanism that, yes, I am the infected cell, or I am the cancerous cell, and the CD8 or the cytotoxic cell needs to come and kill me. So it's a cell recognition phenomena. On this particular picture, you can see the activated macrophages. These are the inactive macrophages, and when they get activated, it's because they are stimulated by either antigenic materials or by cytokinins. Activation of T cells. Again, T cells are the are the cells that are cytotoxic cells, and they will be activated by two signals. One is a helper T cell must confirm binding to the process antigen. So if there is an antigen here and the APC or the dendritic cell has taken in the antigen and broken it down inside, then these fragments of chemicals or molecules are actually released on the site as called as a TCR receptor, and this binds to the MHC antigen complex, and now the APC is stimulated, and the T cells can then come in and attach to it and destroy that particular cell. Sometimes the T cells are also stimulated by cytokinins, which will also help to proliferate and destroy the cell. So here is an MHC infected on the virus. So notice that here is a virus infected cell, and it's got processed antigen. Notice that the processed antigen presented with the MHC1. And the MHC molecule is then going to be recognized by the cytotoxic CD8 cell, and it will cause destruction of that infected cell. So going back, there are two kinds of these MHC complexes. MHC2 that is formed at the beta cell, and it pulls in the T cells towards it, and once a T cell comes and attaches, it releases the MHC histocompatibility complex protein 1. Going back up here to see if I forgot something. No, so pretty much you've done most of this earlier in immunity and Bio 202. This is just a recall of what you did earlier in your biology classes. Now, how does the antibody cell-mediated cytotoxicity works? Now, notice that antibodies can be produced in the humoral immunity and also by cell-mediated immunity. And these antibodies that are formed by this process will actually stimulate the natural killer cells, and the natural killer cells will then go and destroy or perforate in the enzyme. So notice up here you've got the epitopes. The antibody comes in here. It's activated the eosinophil, which is the mast cells, and that releases perforin and lytic enzymes, and these will then damage they're damaged by the macrophages that are coming and attaching to these cells. So this is an organism such as a parasite that are too large for ingestion by the phagocytotic cell, and they must be attacked externally, and that is by the antibodies that it produces. Here is a comparison between both the humoral, that is the antibody-mediated response, and the cellular or the cell-mediated response. So notice here we have the cellular extracellular antigens. These bind to the B cell, for which it is very specific. B cells will either work in cooperation with the T cells to target the infected cells, or the B cells, when sometimes without the effect of T cells, will produce plasma cells, and they will release antibodies against the antigen. So here's a story about how the antibodies are released. On the other hand, if you notice on the right side, you have an exposure to an antigen that is on the surface, and this can be on a virus or a bacterium, and this binds to an APC. This actually binds to an APC, and the T cell binds to there to form a complex, and now this infected cell is going to activate a helper T cell, because it says, well, I'm the infected cell, come and start killing me. The activation of the macrophage will enhance the phagocytotic activity. The CD8 T cell will become the cytotoxic lymphocyte, and it is able to lyse or break down the target cell. We have we went back a little too fast. We have memory cells that are also T cells and B cells, and they are always present to in our system to rapidly encounter if the antigen has been exposed later in life. So this concludes our understanding of the cell-mediated response.